Soil microbiology, the observation of diversity of microorganisms in soil. To begin this experiment, you will need to measure 1 gram of soil. Place your weigh boat on the scale and tare the scale. Now, measure out 1 gram of soil. The 1 gram of soil will be the equivalent to 1 milliliter of a liquid sample. You will now add the 1 gram of soil to 99 milliliters of a peptone solution. Carefully pour the soil into the peptone. Replace the cap. Shake the sample vigorously and allow it to sit for approximately 1 minute. The 1 gram of soil in 99 milliliters of solution has a dilution factor of 10 to the negative 2. You will now use a serological pipette to transfer 1 milliliter of your 10 to the negative 2 solution into another 99 milliliters of peptone. Remove the cap from the 10 to the negative 2 dilution, insert the pipette, and carefully measure 1 milliliter of solution. You will now place that 1 milliliter of 10 to the negative 2 dilution into the resulting 99 milliliters of peptone. Place the used pipette back in the sleeve and remove it from the pump. This should be disposed of in the biohazard. This second dilution will have a dilution factor of 10 to the negative 4. You are now ready to label your plates. You will need 9 plates because there are 3 different dilution factors and 3 different types of media being used. The first row of plates will each contain nutrient agar. You should label the plates from left to right 10 to the negative 2, 10 to the negative 4, and 10 to the negative 5. These will be your final dilution factors. The second set of plates will contain saborodextrose, or SD agar. These should also be labeled from left to right 10 to the negative 2, negative 4, and negative 5. Your last set of plates will have the same 3 dilution factors from left to right, and all 3 of these plates will be labeled GYE for glycerol yeast extract. Be sure to label your plates on the bottom and on the edges so as not to obscure your view of any of the samples that you will be viewing. Now, begin turning your plates over in preparation for adding your samples and pouring agar. To add the samples, you will need both a serological pipette and a micropipette. Carefully flip the plates over so you can easily pick up the lid. From your 10 to the negative 2 dilution, you will remove 1 milliliter of sample. Using the serological pipette, remove the 1 ml and place the sample in the center of the empty plate. Be sure when putting your sample in the plate that each plate is labeled 10 to the negative 2 and that you are obtaining your sample from the 10 to the negative 2 French square. When you have completed this for this first set of plates, you need to change the serological pipette. Place the used pipette back in its sleeve and carefully remove the pump. Using a new pipette, you will now begin adding 1 milliliter of your 10 to the negative 4 dilution to the second column of plates. The same procedure will be used for this as was used for the 10 to the negative 2. Remove 1 ml and place it in the center of the plate. The third set of plates will require the use of a micropipette. For this set of plates, which is your 10 to the negative 5 dilution, you will be adding 100 microliters of sample to each plate. This set of samples is 1/10 the amount of solution as was placed in the others. Therefore, the dilution factor will be 1/10 less. Now that your samples are placed, you are ready to begin pouring the molten agar. You will be using 3 different types of agar: nutrient agar, saborodextrose agar, and glycerol yeast extract. Carefully lift the lid and pour the agar in the plate. The agar should be poured gently. It should also be handled very carefully since the molten agar is very hot. Once the agar has been poured into the plate, you should replace the lid and gently slide the plate in a short figure 8. This will ensure mixing and spreading of the media and sample. Soil plates show great diversity of organisms. In this particular plate, we can see an actinomycete, a mold, a bacteria, and yeast. The 3 different agars being used are each used to promote growth. Glycerol yeast extract promotes actinomycetes. Nutrient agar promotes the growth of bacteria. Sabdex promotes the growth of fungi. Yeast are another form of fungi. Using a simple serial dilution and 3 different types of enriched media, you should be able to observe and quantitate the diversity of different organisms found in soil.